Hello, I am Ju Chen from the Rockefeller University. Today, I'd like to discuss the mechanism of Cftr correction by type 1 correctors. There is no relationship to disclose related to this presentation. Cftr is a single polypeptide containing two transmembrane domains, two nucleotide-binding domains, and the R domain. This structure presented here is determined in the dephosphorylated ATP-free form, presenting a closed channel. Missense mutations are distributed all over the structure. F508 is located at the interface between the transmembrane domain and the NBD1. As this interaction is important to maintain the integrity of the structure, as well as transduce conformational change in the nucleotide binding domain to the transmembrane domains, deletion of F508 will weaken the interaction between transmembrane domain and the NBD, and thus cause folding and functional defects. Many correctors has been developed. They can be categorized into different clusters based on their functional redundancy or additivity. Today's work will focus on the type 1 correctors, Cftr and the Cftr. This is a maturation assay quantifying the relative amount of mature Cftr. As you can see, the addition of either Cftr or Cftr or Lysosomal and Co4A will all increase the relative amount of mature Cftr. We'll also image the location of delta 508 using the green fluorescence tethered to Cftr. Delta 508 superposition well with the ER marker, but not with the plasma membrane marker, indicating it's largely retained in the ER. Addition of Cftr rescued some delta 508 to the plasma membrane. As you can see in those regions, we can see green dots ventured beyond the ER marker. So the question is, does those correctors act on misfolded Cftr to restore its structure, or they stabilize folded Cftr in the lative conformation? The second possibility is supported by several studies from multiple laboratories as listed here. In those papers, data were presented showing that Cftr or S18 binds directly to either purified Cftr, or they can act on delta 508 after it's rescued to the cell surface. So encouraged by those data, we also performed direct binding assay using a radioactive labeled Cftr. In this assay, we can see the binding of Cftr is increased as we increase the concentration of Cftr with a KD of 8 nanomolar. A control protein, which is closely related, showed no binding. We also used the competition assay to demonstrate the Cftr can compete off radioactive labeled Cftr with a Ki very close to its measured KD. And the Cftr also competes with Cftr with a Ki of about 115 nanomolar. The other two correctors, Lysosomal and Co4A, cannot compete with Cftr. So with this information, we went ahead and determined the structure of Cftr in complex with Cftr in both ATP-bound conformation and dephosphorylated ATP-free conformation. In the absence of ATP, the structure was determined to about 4 angstrom, and a prominent density is observed at that is kind of consistent with the shape of the Cftr structure. This density is absent in the map that's obtained in the absence of the drug, which is shown here. A higher resolution structure, 2.7 angstrom, was determined using phosphorylated Cfdr in the presence of ATP. In this case, you can see high quality density that fit very well with the chemical structure of lumacaftor. So the binding site is located at the interface of the membrane and the cytosol, which I'm showing it here. So this is where the lumacaftor will bind. What highlighted by arrow here is the potentiator isocaftor. So binding sites of lumacaftor and isocaftor are quite far apart from each other, consistent with those two drugs can use together. And the binding site is actually have a large pocket on the transmembrane domain 1, and half of the molecule of the drug is buried inside this pocket. The other half extend outside, interacting with the more hydrophilic region of TM1, and they also exposed to the lipid head group. Previous cell-based studies showed that the minimum domain sensitive to lumacaftor is the N-terminal 375 residues. Removing 5 residues, 371 to 375, makes this drug ineffective. And this region is highlighted here. As you can see, although these residues do not directly interact with the drug, they interact with the N-terminal region of transmembrane helices 1, and together they tether the two parts of the protein together and form a part supporting a structure of the binding pocket. So those observations also means that the three-dimensional structure of the binding site must form for lumacaftor to act on, indicating that lumacaftor binds to TMD1 in its folded state. Next, we determined the complex structure with tezacaftor. Consistent with the binding competition assay, tezacaftor also binds to the same site, and the density is very convincing, fits tezacaftor specifically. Here, we superimpose the two drugs, lumacaftor in lighter yellow and the tezacaftor in orange. And you can see the region that they share, we call the BCC head group, which is highlighted here, also highlighted in the two schematic views in gray, inserts into the pocket between the transmembrane helices, and they're also interacting with the same set of amino acid inside the pocket. And the other half of the drug have a slightly different conformation and interacting with different groups of amino acid, particularly lysine 68, which I will come back to it later, forms a strong salt bridge with lumacaftor, but this residue does not interact with lumacaftor at all, with tezacaftor at all. Because the structure of the drug complex are determined using folded wild-type Cftr, we asked if this very binding site is the site of action during Cftr folding. To answer this question, we introduced the mutations at the binding site to the delta 508 background and analyzed its ability to respond to the correctors. So one particular example I want to show here is leucine 195. Leucine 195 is underlining the pocket, form very close interaction with tezacaftor and the lumacaftor. So a substitution by tryptophan will make this pocket narrower and cause stereo occlusion of the drug. And as you can see from the gel shift assay, the leucine 195 tryptophan mutations specifically reduced the response to both tezacaftor and the lumacaftor, but not to the control cocktail of elezacaftor and CoL4A. And these gels will run many times, and then quantification was shown here. So this is a delta 508 normalized to 1. Then LUMA-Cftr and TAZA-Cftr do not rescue the mutant, but the other non-type 1 correctors do. Confocal microscopy also showed that LUMA-Cftr cannot rescue the mutant, but the other correctors can. As you see, the Cfdr superimposed with the ER marker in the presence of LUMA-Cftr and very little expression on the cell surface. So we made several of similar mutations, including alanine to Y mutation and the serine to F mutation, and consistently see reduction of response to type 1 correctors, but not to the control correctors. Next, I want to talk about lysine 68. As I mentioned, this residue only interacts with LUMA-Cftr. So in the gel shift assay, we also see the K68I mutation does not respond to LUMA-Cftr, but it can be rescued by TAZA-Cftr, indicating specificity conferred by this residue. So I hope by now I can convince you that type 1 correctors bind to Cftr in the folded conformation. Then the question is, how does this interaction help folding? The cavity observed on TMD1 reminds us about early work of Brian Matthews. He created the cavities in lysozyme, determined the structure of a series of mutations, and also measured their stability. What he found was quite remarkable. This plot shows the internal cavity destabilized protein. Here is the destabilizing energy determined by the mutation, and here is the cavity size introduced by the mutation. And there's almost a linear relationship. The larger the cavity, the more destabilization it shows. And notice this is a log scale. So the cavity we see in Cftr that's being filled by the drug is about 360 cubic angstrom, which is beyond the scope of this plot. So having such a cavity inherited in Cftr will destabilize the protein substantially. That's why even native Cftr, wild-type Cftr is quite unstable. Secondly, what we see is from work of Steve White and Gunnar Wanhani, they developed a method to measure the energy of membrane insertion based on the sequence. Cfdr has many polar residues in the transmembrane region, and we used the same method developed by Gunnar and Steve to calculate the energy of membrane insertion of all 12 helices. And you'll notice several of them are positive, especially the 4 helices, transmembrane 1, 2, 3, and 6, all have a positive delta G, which means those helices are very unstable in the membrane. The drugs, LUMA-Cftr and the TAZA-Cftr, both are hydrophobic and they are happy in the membrane. So by binding to those 4 helices, they can stabilize the helices in the membrane environment, also contributing to the ability to stabilize the protein. Lastly, but not least, the work from Bookman's lab has shown that Cftr folding occurs mostly co-translationally. So as the peptide emerging from the ribosome, TMD1 is synthesized first. So it becomes partially folded in the membrane, waiting for the rest of the protein to be made. Putting all this information together, we propose a model that could explain a lot of data in the literature why the type 1 correctors can improve Cftr folding. Once the N-terminal transmembrane domain 1 is synthesized, it adopts a tertiary structure that is intrinsically unstable in the membrane with positive delta G of 4 of his helices, and the other 2 helices needs to pack against the C-terminal region of the protein. And the binding of Cftr or Cftr stabilize TMD1, making it less susceptible to targeted degradation by the protein quality control machinery. The Cftr folding is a highly cooperative process. Stabilizing TMD1 would ultimately increase the overall probability of forming a fully assembled structure and thereby allosterically rescue many different mutations. Some of them are like delta 508 that's located in completely different parts of the protein. This mechanism is also consistent with the synergy between Cftr and the suppressor mutations. Cftr extends the lifetime of TMD1, and the mutations stabilize other parts of Cftr or enhance the interdomain assembly. So together, they can achieve higher rescuing efficiency. With that, I'd like to thank particularly Carol, who did all the correctors work I presented. And the Cftr project was established by amazing postdoc Zhe Zhang and equally amazing graduate student Feng Yu Liu. Both of them moved on now. And currently, Jasper and Carol are continue the project. And this picture was taken before COVID, and this one is taken just now, so you will all can see what happened to us. And I thank HHMI and the CF Foundation for funding. Thank you very much.
Good morning. I'm glad to be here and share with you our recent study on the roles of ATP-binding site 1 in Cftr's functional stability. I am currently supported by a fellowship from AbbVie. But before the structures of Cftr came out, people imagined that its structure and gating must closely resemble an ABC transporter. Structurally, an ABC transporter has 2 transmembrane domains and 2 nucleotide-binding domains. The protein transits between an inward-facing conformation and an outward-facing conformation. This is the so-called flip-flop motion. In the inward-facing conformation, the nucleotide-binding domains, or the NBDs, are separated. Binding of ATP to the 2 ATP-binding sites triggers dimerization, which powers the conformational changes to flip the protein to this outward-facing position. When ATP is hydrolyzed, the dimer interface breaks apart and the protein resets to the inward-facing position. And since Cftr is a member of ABC transporters, the 2 conformations were thought to represent the closed state and the open state. ATP-induced dimerization controls the opening, and hydrolysis controls closing of the channel. Importantly, hydrolysis only takes place in the second nucleotide binding sites. Cftr's site 1 does not hydrolyze ATP, and therefore it's known as the degenerate site. The simple idea that the ABC transporter's inward-facing conformation corresponds to Cftr's closed state, and that the outward-facing conformation corresponds to open state, is getting support from Dr. Zhu Cheng's recent cryo-EM studies. The dephosphorylated ATP-free structure indeed shows an inward-facing conformation with a closed pore. The phosphorylated ATP-bound Cftr structure shows an outward-facing conformation, although the pore needs to dilate a bit further to allow chloride passage. It is believed that this dimerized structure is already very much like an open channel. Now it seems that gating of Cftr indeed resembles ABC transporter's flip-flop motion. The TMDs and MBDs move as a rigid body. The closed channel has widely separated MBDs, and the open channel has dimerized MBDs. This gating model sounds nice and clean, but is it really that simple? In fact, Previous studies suggest a different scenario. It is generally agreed that an open channel has a dimerized MBD. However, both ligand exchange experiments and mutant cycle analyses suggest that after Cy2 hydrolyzes ATP, the channel closes, but the ATP at Cy1 remains bound, and the two MBDs remain partly engaged. Biochemical studies also support that, unlike the hydrolytic product in Cy2, which is released upon hydrolysis, ATP in Cy1 stays for tens or even hundreds of seconds. Before the Cy1 ATP dissociates, it is possible for another ATP to hit Cy2, and the two MBDs can redimerize and channel opens again. So in this scenario, Cftr's MBDs should not experience full separation for very long because this Cy1 ATP would help the two MBDs remain connected. But we do not have direct structural evidence to validate this closed state. In fact, cryo-EM data support a closed state with separated MBDs. But bear in mind that the cryo-EM closed channel is a dephosphorylated structure. It remains a question whether a phosphorylated Cftr would also have that widely separated MBDs. But to understand the structure-function relationships of this closed state with fully separated MBDs, we ask a simple question. What would happen if we force ATP to dissociate from Cy1, resulting in a complete separation of phosphorylated Cftr's MBDs? And as described earlier, ATP can remain bound at the degenerate Cy1 for tens of seconds. So one simple way to force MBD separation is to remove ATP for a prolonged period of time. We use the patch clamp technique to record the activity of Cftr in an inside-out configuration, which allows us to apply and remove ATP. Here is a recording of 3 phosphorylated wild-type Cftr. Upward deflection represents opening. In the presence of ATP, you can see 3 channels opening and closing in a stochastic manner. When ATP is removed, all channels close. Shortly after 10 seconds, we add ATP back. All channels come back almost immediately, and there is no distinguishable difference in the activity compared to the previous section. However, if the channels do not have ATP for a long time, here for 180 seconds, and we reapply ATP, only one channel opens right away. You need to wait another many seconds until you see a second opening, and you need to wait even further before all 3 channels reach the activity they previously had. This graded recovery is very different from what we've seen with 10-second washout. It seems that after prolonged absence of ATP, the channels need longer time to recover. It is as if the channels can take a short nap and get right back to work. But when the channel sleeps for too long, it has a harder time waking up. So simply put, Cftr does not like to be in this condition long enough for its ATP to dissociate from the degenerate side. And this problem with slow recovery is even more obvious with macroscopic currents. Here are wild-type channels. We remove ATP for increasingly longer durations and see how the currents recover. First, following a 5-second ATP washout, nearly all currents come back to the steady state immediately when ATP is back. Similar fast response after 10-second washout. However, at 30 seconds, you start to see a clear biphasic response. This second slow phase becomes larger when ATP is absent for much longer. If we fit the curve with a double exponential function, we get 2 time constants. This biphasic course suggests that after prolonged ATP washout, there are at least 2 populations of channels. One reacts to ATP rapidly, the other somehow reacts slowly. And this time constant 2, or tau 2, reflects the speed at which the current recovers in this second phase. And as you can see, with longer washout, this tau 2 gets larger, meaning that the current recovers slower. And to understand what it means to have two populations of channels, let me use this bar to represent the maximum current, and the different shaded blocks represent the fraction of the fast and slow response. As we remove ATP for longer and longer, the fraction of the slow responders becomes larger. And we hereby define this transition from the fast to the slow group rundown of Cftr. Rundown channels become less sensitive to ATP. However, despite their slow response, they at least manage to recover. You can see at the end of the recording, if we remove ATP briefly, all channels can again rapidly come back, meaning that those who had experienced rundown are now recovered to normal, fast responding channels. So this rundown is reversible. And now please pay attention to the raw trace again. If this dotted line is the maximum current we have on this patch, you'll notice that there is a permanent loss of current after the 1/82nd ATP washout. The total current does not reach its original level, and this is not due to dephosphorylation, as pKa does not help. A larger decrease happens after a 300-second ATP washout. In contrast, if we remove ATP briefly, like the arrows in the beginning and also at the end of the recording, the currents did come back to its prior level. So these observations suggest that there is an irreversible loss of currents after prolonged washout of ATP. To this point, we have two observations that are time dependent. First, the longer the washout, the greater this second slow phase in proportion to the total current, namely the reversible rundown. Secondly, the longer the washout, the larger this permanent loss or the irreversible rundown. So to use our previous analogy, it seems that when the channels sleep in ATP-free solution for a long time, some of them, those that did not run down, wake up quickly. Some of them feel kind of groggy and take longer to fully get up. That is, channels undergone reversible rundown. And still others just never open their eyes again. So you have irreversible rundown. The key point here is that rundown on wild-type channels, both reversible and irreversible, happens only when ATP is removed for a long time. Rundown does not happen when the channels have enough ATP. As shown here, the wild-type Cftr currents in ATP remain very stable for at least 30 minutes. Also, for channels to run down, it has to be in a continuous absence of ATP. Here we repeat a 10-second washout of ATP for 30 times, and each time the current comes back to the maximum level. You don't see any rundown. The cumulative time when ATP is absent is 300 seconds. But if we put the channels in a continuous absence of ATP for 300 seconds, we see significant rundown. And here's how we interpret these results. The closed channels in the short interval, as indicated with the black triangle, are different from the closed channel in the prolonged washout, colored in red. In the short washout, the channel is closed, but Cy1 ATP is still bound. In the long washout, Cy1 is empty. We can therefore say that rundown happens only when the MBDs are fully separated. If so, we would expect that as long as Cy1 has ATP, the channel does not run down. To test this hypothesis, we repeat a similar protocol on wild-type channels, but this time, instead of removing ATP, we keep the channels in 10 micromolar ATP for different durations. From previous studies, we know that 10 micromolar is sufficient to keep Cy1 ATP constantly bound, but not enough for Cy2. Here you can see that although most channels are closed in 10 micromolar ATP, as reflected by a drastic drop of the macroscopic currents, they all come back rapidly when 2 millimolar ATP is supplied, suggesting that there is minimal rundown. In contrast, complete removal of ATP caused severe rundown. This result is consistent with our hypothesis that holding Cy1 together prevents rundown. If this idea is correct, when binding of ATP in Cy1 becomes weaker, rundown should happen more often. We can make mutations to test if this is true. The K464A mutant in Cy1 destabilizes the electrostatic interaction for ATP binding, so the affinity for ATP in Cy1 becomes lower. Here are the K464A channels. It clearly shows that a very large fraction of current is lost after ATP washout, meaning that the irreversible rundown is much worse than wild type, which would have just decreased by 20% in the same condition. From this observation, it follows that making a tighter connection in Cy1 should prevent rundown. To test that, we replaced ATP with an analog phenyl ATP, or PATP, that is known to bind tighter to the MBDs with its extra phenyl group. Here, with PATP, the currents come back to almost the same level after long washout, meaning that there is little rundown. We then conclude that PATP, which gives a tighter binding in Cy1, indeed prevents rundown. Another mutation, W401G, which also destabilizes ATP binding in Cy1, shows similar results. The conclusion is that stable binding of ATP in Cy1 prevents rundown. So our very first question was, what happens when the MBDs of a phosphorylated Cftr separate? Now we showed that the channel would run down easily, and the rundown can be prevented by stable binding of ATP in Cy1. Here, let me propose a model to describe the potential mechanism for rundown. The moment ATP is removed, the channels close upon hydrolysis in Cy2, but ATP in Cy1 stays longer. This closed state with partially connected MBDs, labeled C0, is able to bind the next ATP immediately and opens right away. However, when the channel remains in the ATP-free environment for long enough, the channel first closes upon hydrolysis, but then ATP in Cy1 eventually dissociates and the MBDs fully separate. To open again, the channel has to bind two ATPs and redimerize. This might be the reason why the channel responds to ATP much slower, a result of reversible rundown. And based on the observation that longer ATP washout results in slower recovery, as I showed you earlier, we propose that the separation of MBDs might be a continuous process. To what extent the MBDs separate relatively to each other remains a mystery, but there might be multiple states where MBDs have different degrees of separation. Furthermore, the distance between the two MBDs might eventually grow so large that dimerization becomes very difficult, making the backward reaction practically irreversible, hence irreversible rundown. We are aware that without the atomic structures, it is hard to tell exactly what a rundown state looks like. Nevertheless, we hope to convey the idea that the opening and closing of Cftr may not be as simple as a rigid body motion. In fact, Cftr behaves poorly if we allow its two MBDs widely separated, as happened in the ATP-free apool condition. In other words, Cftr is not happy with its two MBDs fully separated. So to summarize, we want to emphasize the importance of the degenerate ATP binding site. We have long wondered whether the loss of hydrolysis ability in Cy1 brings any kind of advantage to the channel, or is it just an incident in the history of evolution and the channel lives with it? Now, we perhaps can say that by losing the catalytic activity, Cy1 is able to keep ATP bound and the two MBDs close enough, preventing it from going to the rundown state. Cy2 by itself may be sufficient to gate the channel, but it is really this degenerate Cy1 that makes sure that the channel is able to respond to ATP efficiently and timely. Finally, I would like to thank all lab members in Dr. Huang's lab who helped with this project and the co-authors of this recently published paper. And thank you all for your attention.
Hello, my name is Lars Platte, and I'm an assistant professor for chemistry and biological sciences at Vanderbilt University. First of all, a big thank you to the organizers for inviting me to the symposium on modulating the Cfdr molecule. So I'm excited to share with you on how we use interactome profiling of Cfdr misprocessing variants to probe their divergent theratypes. I have no relationships to disclose related to this talk. The underlying cause of cystic fibrosis is a defect in the function of the chloride bicarbonate transporter, cystic fibrosis transmembrane conductance regulator, in short Cfdr, shown here on the right. Patient mutations in Cfdr are generally classified based on their molecular defect. And this has been, of course, very useful for addressing these distinct defects with specific therapeutic approaches. Class 2 misprocessing mutations make up by far the largest patient group and will be the focus of my talk today, and specific examples of the mutations are listed here. These Cfdr variants are misfolded and then prematurely degraded instead of being trafficked to the cell surface of epithelial cells. As for all cellular proteins, Cfdr biogenesis and trafficking is tightly regulated by a myriad of protein-protein interactions with the cellular proteostasis network. I want to briefly take the time to walk you through the different stages. As the protein gets translated by the ribosome and co-translationally inserted into the ER membrane, chaperones like HSP70s and HSP90s and a variety of co-chaperones assist with the folding. If the protein then passes several quality control checkpoints, it can engage with trafficking factors to translocate through the Golgi, where it gets further modified with glycans, and then to the plasma membrane. On the other hand, misfolded Cfdr states are recognized by proteostasis factors and instead rerouted towards degradation machinery, in particular E3 ligases that ubiquitinate Cfdr, which is then degraded by the proteasome. So as such, cystic fibrosis is a loss-of-function protein misfolding disease. Proteostasis factors also play a role in clearance of Cfdr from the plasma membrane through endocytosis and then targeting to the lysosome. In my research group, we study the coordination of the proteostasis network interactions using mass spectrometry, and our goal here is to apply these tools to elucidate the quality control mechanisms for Cfdr. Several drug molecules have revolutionized cystic fibrosis therapies and can address the specific molecular defects. So I'm highlighting potentiator molecules such as ivacaftor or VX770, which improve the gating and conductance function of Cfdr. On the other hand, a variety of corrector compounds have addressed the folding and trafficking defects. I've shown here the chemical structures of several of these correctors on the right, and importantly, VX809 or lumacaftor was the first generation of FDU-approved correctors. Next generation, VX661 or tezacaftor shown below is a structural analog of 809. And then elexacaftor or VX445, as shown below, is the latest approved corrector that is part of the Trikafta triple combination therapies. While all of these correctors restore processing of mutant Cfdr, One important point to highlight is that the Cftr misprocessing mutants don't all respond to the same degree to these drugs. This is described as having distinct theratypes. So if we take delta F508, for example, the most common Cftr patient mutations, lumacaftor is quite ineffective at restoring the levels of band C Cfdr, which can be taken as a measure of complex glycosylated traffic protein. Compare wild-type Cfdr here on the left. On the other hand, other class II mutations respond much more strongly to the corrector molecule. One well-studied variant is P67L, where the mutation occurs here in the N-terminal lasso region. When treated with VX809, mature Cfdr levels of P67L are almost fully restored to wild-type levels. And importantly, the divergent theratype between 508del and P67L is also reflected in electrophysiology measurements, where channel function is restored by only about 10% compared to 70% for P67L. There are other mutations that exemplify this divergent theratype to 809 corrector, for example, G85E, which responds very poorly, while L206W is strongly restored. We can classify these mutations as hyper-responsive to the drug or poor responsive on the right. Of course, this now raises the question, what factors determine the divergent theratype to the pharmacologic correctors? These correctors act as pharmacologic chaperones binding directly to Cfdr. So on the one hand, they provide some structural stabilization and can influence the thermodynamics of folding and also membrane insertion. Excitingly, the recent VX809 and 661 bound cryo-EM structure from Jui Chen's team gives us a better insight into this structural stabilization. It turns out that the binding site is actually very close to the lasso region where P67 is localized, so one could speculate how direct molecular interactions with the lasso region could specifically rescue P67L. But on the other hand, of course, we need to consider all of the proteostasis network interactions that regulate the processing of Cftr within the cells. We know very well from prior mass spec studies that these interactions are elevated across the board for delta F508 compared to wild type. And so this highlights the increased protein quality control surveillance of the mutants. The same studies have also shown that the rescue conditions, low temperature or correctors, attenuates the proteostasis interactions. And of course, finally, these altered proteostasis interactions shape the conformation of Cftr, for example, through specific chaperone interactions, which can then in turn influence drug binding. This complex interplay between corrector binding and proteostasis network remodeling motivated our research question. We asked, what are the changes to the Cftr proteostasis interactome that mediate the response to the correctors? To address this, we turn to liquid chromatography mass spectrometry as our technique of choice to quantify these changing protein interactions. We then organize the interactions into particular proteostasis pathways and look at which ones are distinctly altered by VX809. And with that information, we validated the importance of some of these pathways, as I'll show you. Now is a good time to introduce Carleen Sebusap, who was the main driving force behind this project and was brave enough to join my new research group as my first postdoc. Carleen was a PhD student in Eric Sorsha's group at Emory, where she had carried out the initial characterizations of P67L as a hyperresponder. And so she brought a lot of the Cfdr expertise to my new group. And Carleen collaborated with Eli McDonald, a very talented graduate student in my group who's now carrying the project forward. I want to take a moment to introduce now our quantitative mass spectrometry technique to you because it is unique in that we can multiplex particular Cfdr variants and drug conditions to directly compare the interactions. We begin with cells that express different Cfdr variants, wild type, poor and hyperresponsive mutants, and we can then treat the cells with mock or with the corrector drugs. Then we immunopurify Cfdr and its interaction partners using antibody conjugated beads. And the purifications are shown here on Western blots, where you can clearly see the divergent restoration again of band C levels. Then we can use quantitative mass spectrometry with tandem mass tagging to analyze all of the proteins that are found in the samples. The TMT tags contain heavy isotopes in different spots on the molecule and act essentially as barcodes for the peptides, allowing us to combine samples and then deconvolute the peptide abundances after collision-induced dissociation of peptides. We can then pool all of these peptides and analyze them by two-dimensional liquid chromatography coupled to tandem mass spectrometry. Subsequently, we can identify all of the peptides but after collision-induced dissociation, and this also liberates specific TMT reporter ions that now carry the quantitative information that allows us to compute the relative amounts of these proteins in up to 11 different conditions. To filter out Cfdr interactors, we first compared the abundances between Cfdr expressing cells and mock cells that don't have any Cfdr. In the volcano plots, we show the enrichment of proteins on the x-axis and the statistical significance on the y-axis. And importantly, we saw a very clear enrichment of all of the Cfdr variants in our study. We then validated that we could also detect interactors from previous studies, in orange here, but we also found new interactions. So next we grouped the Cfdr interactors by particular proteostasis pathways, and we also looked at the overlap with prior Cfdr interactome studies. And importantly, here we found comparable overlap, and we identified many of the core factors with validated roles in Cfdr processing and function. So some of these I've highlighted here with red arrows. Next, we were interested in comparing the P67L interactome to delta F508. So for this analysis, we had to normalize the interaction fold changes to wild-type Cfdr to provide a common reference point. And then we also aggregated individual protein changes into related pathways, for example, shown here, interactors related to translation. For delta F508, shown here in blue, it's well established that interactions are elevated compared to wild-type. And so we see the same elevation for P67L with translation proteins, here in green, and also compared across the board with all of the other pathways that we've prioritized. Overall, then, the two misprocessing mutations exhibit a similarly remodeled interactome. These stronger interactions with protein quality control machinery are perhaps not too surprising given the common defect. So far, I just showed you data without the corrector drug. But the key question was, of course, what happens to the interactome when we add VX809, and what changes dictate this divergent corrector response? So through our multiplexing, we could directly compare the interaction changes in the same experiment now. I'm going to show you the same aggregate interaction changes by pathway. Again, I would like to stress that these are relative to wild-type levels, indicated by the dashed line here. And then in gold and purple is now shown what happens to the interactions when VX809 is added. For both variants, we see that the interactions with translation factors get reduced closer to wild-type. This also happens with folding machinery, such as chaperones. But then when we go to the proteasomal degradation, the changes start to diverge. We see a lot less resetting of these interactions for delta F508 and more for P67L. And this is even more pronounced in the subsequent pathways, autophagy, trafficking, and endocytosis. A key observation is that we see a divergence post-folding. So subsequent pathways remain engaged with delta F508 while they reset for P67L. We also looked more closely at specific protein factors that may play a role here. One protein that stood out as being differentially impacted by VX809 was the co-chaperone DNAJB12. This was actually revealing because of prior studies showing that DNAJB12 triages proteins from folding towards degradation, so right at this step. You just saw data for P67L and delta F508, but how generalizable are these observations to other mutants? So for this purpose, we also looked at L26W as another hyperresponder, and G85E, which is completely non-responsive. We saw similar attenuation of protein interactions across the board for the hyperresponsive L26W with all pathways. On the other hand, G85E was completely refractory to VX809 and maintained the same high interactions across the board. This was not just with the later pathways, but interestingly also with translation and folding. We saw that the divergence between hyperresponsive and non-responsive variants occurred when degradation pathways became engaged. So therefore, we became interested in following up to probe how proteasomal and autophagy degradation pathways could contribute to this distinct correction. For this, we turned to small molecule inhibitors. We used bortezomib for proteasome inhibition, and with this, we see, of course, a buildup of band A and B, which correspond to ER-localized Cfdr. For delta F508, we also see a little bit of restoration of trafficking reflected as increased band C, and this is consistent with prior data for proteasome inhibition. But when we now treat it with the combination of corrector and bortezomib, we see additional band C for delta F508, meaning that the correction can occur when proteasomal degradation is inhibited. But this was not the case with P67L. Here we no longer observed restoration of band C, indicating that proteasome degradation is actually required for correction. Next, we also looked at lysosomal protein degradation using E64 as an inhibitor. And here we see very little effect of the inhibitor on P67L correction, telling us that the correction is insensitive to autophagy. So let me now summarize our findings so far briefly. We saw that early proteostasis interactions with translation and folding machinery are being reset with VX809, which is consistent with the corrector binding co-translationally to Cfdr, so very early on. The only exception here is G85E, which is entirely unresponsive to the corrector. But then subsequently, the paths diverge. VX809 reduces the routing towards degradation pathways for the hyperresponsive variants, meaning that they are mostly present in corrected conformations that are now trafficking competent. On the other hand, delta F508 does not pass these quality control checkpoints and remains routed for degradation, or it also remains trapped in the secretory pathway, which could explain the increased associations that we see with trafficking factors. Lastly, we saw that active proteasomal degradation was necessary for the correction mechanism of the hyperresponsive variants. So this may indicate that some non-correctable conformation is still present and require proteasomal clearance to not disrupt trafficking of the corrected subpopulations. So far, I've shown you data for VX809 or Cftr, which was the first FDA-approved corrector molecule. But you may wonder, what about the next generation correctors, in particular Cftr or VX445, which is a key component of the Trikafta triple combination therapy, and because it can much better restore delta F508 trafficking and its function. This, of course, raises the question whether VX445 similarly exhibits distinct theratypes towards other Cfdr variants. One of my students, Min-Soo Kim, decided to tackle this exact question. I'd like to give a short plug to Min-Soo, who's presenting in more detail on the Interactome remodeling in response to Cftr. So he's presenting in the Thursday afternoon session listed here, and he also has a poster. So I highly recommend that you tune in for some more details. I am just going to briefly summarize Min-Soo's data here, which was quite exciting because, again, P67L actually showed a divergent response to VX445. You can see here how it is corrected by 809 and also its analog 661, but VX445 is not effective at restoring P67L band C levels. And this is in contrast to L26W, which is very well corrected by VX445. This is also mirrored in a functional measurement of short circuit current recordings conducted by our collaborators. And here you see nice restoration of Cfdr conductance with 661, but to a much smaller degree with 445 at different concentrations. This, of course, again raised the question, what proteostasis factors mediate this divergent response? So Min-Soo also carried out the Interactome profiling and actually discovered that VX445 does not reset even early translation pathways for P67L. And again, also degradation pathways are differentially impacted by these drugs. Min-Soo is now following up on some of these hits to identify distinct factors that may be important for the response. In conclusion, I've shown you that Cfdr mutants exhibit divergent theratypes to these corrector drugs. Not all of the drugs are similarly effective at restoring Cfdr processing, and this of course has implications for their therapeutic use. Cftr is approved now for over 90% of patients, those who are delta F508 homozygous or heterozygous in combination with over 100 variants. For this reason, it remains important to learn how these different Cfdr variants respond to the drugs and what mechanisms dictate their correction. Our multiplex interactomics here is a powerful tool to profile the distinct proteostasis mechanisms at play here. And therefore, the last point I want to highlight is that we also think that the multiplexing capabilities could be useful for actually predicting corrector theratypes for other rare mutations in the future. With that, I'd like to thank my fantastic research team in the lab. In particular, I've already highlighted Eli, Carlene, and Min-Soo. I would also like to thank our collaborators at Emory University and, of course, our funding sources, in particular the NIH and the Cystic Fibrosis Foundation. And lastly, I would like to thank all of you for tuning into this talk.
Hello, everybody. My name is Andrei Sivachenko, and I work at Therapeutics Laboratory of the Cystic Fibrosis Foundation. And I'm going to talk today about using our theratyping dataset for querying structure-activity relationships in Cftr. I have no relationships or conflicts to disclose. So I'm going to start with describing our theratyping project, what it is, why theratyping, also experimental platforms used, and the type of data collected before we move on to consider how to look at our results. And it's important to realize that a very large number of different mutations, over 1,500 in fact, were reported to date in Cftr gene. And many of those are still poorly characterized. In addition to their sheer number, most of those mutations are extremely rare, down to literally a single known person, which makes running classical clinical trials impossible. And the drugs currently approved for people with cystic fibrosis are effective and life-changing, but the drug can be currently prescribed to people having at least one F508 del allele, which is by far the most common disease-causing variant, though, or also to people who have at least one of 182 additional specific mutations in Cftr. It is very important that while the original FDA approval was obtained after a clinical trial, the label extension to additional and much rarer mutations was approved by FDA based solely on in vitro data. It was literally a combination of previous approval with clinical data showing drug safety and tolerability, and new data obtained in cell cultures in the laboratory, demonstrating that those additional Cftr variants do exhibit improved ion conductance in response to the approved drug. And in fact, that was actually a noticeable shift in the regulatory landscape itself. So thus we decided to initiate this type of project, theratyping project, where we would perform in vitro testing of the existing approved drugs on as large of a set of different known Cftr variants as possible. And the ultimate goal and hope, of course, is to identify additional Cftr variants, no matter how rare, showing response to the current drugs, and thus to enable access to the treatment for more people with cystic fibrosis. Here's how the project was organized. The foundation sponsored the studies conducted at 3 different sites, one being the foundation's own lab, plus 2 academic laboratories. And you have procured the collection of over 650 plasmids, each carrying Cftr cDNA with one of the variants selected for the study. Each lab would independently and transiently transfect those plasmids into FRT cells on site and measure the response to the treatment. And finally, we, in fact, ran 2 studies, essentially identically designed using the same 650 plus Cftr variants, where the first one we measured the response to lumacaftor and ivacaftor, and then the second we measured the response to the most recently approved combination of elexacaftor, tezacaftor, and ivacaftor. And I would simply refer to them hereafter as study 1 and study 2. So some basic statistics for the Cftr variant selected for this study. Again, since we are using cDNA in our experiments, we cannot possibly test all those 1,500 mutations. We can only look at variants located in the exons. We cannot assess any kind of variants located in the introns, promoter regions. We cannot assess variants affecting splicing, causing premature termination codons. So with those restrictions, we obtained the most comprehensive list of over 650 such variants described in Cftr gene from Professor Gary Cutting at Johns Hopkins. And the diagram here shows that most of them are, in fact, missense substitutions, while a relatively small number are in-frame insertions or deletions or complex variants where multiple changes of the gene DNA occur on the same allele. Now, in order to assess the Cftr function in the presence of those variants, we run functional electrophysiology assay. So first, FRT cells are transfected with plasmid carrying a particular mutated Cftr cDNA, and we seed those cells on the transfer filter. Then we give them 4 days to form a layer with tight junctions between the cells. So CFRT is an ion channel for chloride. So at this point, if Cftr is functional or partially functional, there will be some conductance, ion conductance through those channels. And if Cftr protein doesn't work or doesn't get to the membrane, then there will be no conductance because ions are not supposed to go around the cells through the tight junctions. That's about it. So in each of those 2 studies, we would add the corresponding corrector right here, day 4, and just give them those cells another day, give those correctors another day to try fixing any folding and trafficking issues that the mutated Cftr protein might have. And then we perform the actual measurement of the ion conductance on day 5, where we acutely add ivacaftor during the measurement time window. So with such larger number of variants to test, we did employ 24 well plates and tech instruments to perform the measurements in parallel. And here is the standard layout of our 24 well plates, where it's not particularly important here. We just have 4 different Cftr variants measured at once on a single assay plate. Each of the 3 sites would have an assay plate like that, and 2 variants at two wells, two copies, 2 replicates for each variant at each lab would be measured with corrector pretreatment, as I described before, and or without it. Okay, and there are some controls on those wells. Again, only those that are of interest to us are only wild type wells. What is important is the structure of the typical readout from a Cftr variant as shown on the right. Just note that we have here 2 conductance time courses drawn for convenience on the same plot, but originally, of course, each of those would come from its own assay well. So the yellow symbols in this example correspond to the sample pretreated with corrector, whatever it is for the particular study, and the white ones show the signals from the corresponding untreated sample. We start with measurement run. We start the measurement run with inactive Cftr channels, so the conductance is 0. Even fully functional Cftr protein requires phosphorylation for its activation. So after establishing the 0 baseline, we add forskolin, a compound that promotes Cftr phosphorylation. And we see in this example that after this attempt at activating Cftr, we immediately see some conductance in the untreated sample. The signal is the residual activity of the particular variant we are looking at. Of course, it should be compared to the wild type Cftr signal from the control wells, which are not shown here. This residual activity might be comparable to wild type, indicating that the particular variant is not disease-causing, or it might be very low or even 0, of course, for the mutations causing complete or almost complete loss of the Cftr function. In this example, we see that the variant is retaining certain residual function. And in the pre-treated sample, corrector pre-treated sample, the correctors apparently succeeded in bringing more of that partially functional protein to the membrane, so the residual function already becomes higher. And after establishing the residual activity level, we add the potentiator for the full treatment. It's corrector plus potentiator. We usually expect that whatever amount of the protein we get on the membrane and whatever residual function it might have, increasing its open probability should further increase the conductance. And this is exactly what we see, at least here in this example, for both treated and untreated samples. Upon adding ivacaftor, the conductance further increases. And towards the end of the measurement window, we add inhibitor of Cftr, and we of course expect that after that, the conductance drops back to 0. And we certainly extract all those different response levels and other metrics from our conductance traces, and those are what passes for phenotypes in this simple cell-based system. Now, for the discussion of those responses, the numbers of variants found to be responsive, non-responsive to the treatments, how treatment with lumacaftor plus ivacaftor versus triple compound, how they are different. I have to refer you to watch the presentation by Dr. Biller and Dr. Clancy, because what I'm going to discuss in this talk is somewhat an unexpected turn that we realized we could take with this data. So indeed, we have some protein phenotypes, all those functional measurements. And in fact, Western blot data, CNV bands as well, observed after 650 plus different perturbations using the same assay, the same protocol. And the diagram at the bottom shows just the locations of all the studied variants along the Cftr protein sequence. You can see that we have very dense and nearly uniform distribution of our probes. We can try asking questions about associations of those different protein phenotypes with the types of substitutions, with their specific locations on the protein chain, and of course, most importantly, on the 3D structure of the protein. And the 3D cryostructure of the Cftr protein, as shown on the right, is a pretty complex fold with two membrane spanning domains, MSD1 and MSD2, shown with lighter colors, coming together to form the channel, and two nucleotide binding domains that sit inside of the cell under the membrane, NBD1 and NBD2, shown with darker colors. They are also coming together in the fold, binding ATP molecules and controlling opening and closing the channel. Here we just map all those 650 variants in our data set again, but just on the protein 3D structure, just to illustrate one more time that we have the structure very densely covered. Okay, we have representation. Our mutation probes are pretty much in all structurally important regions: membrane spanning, nucleotide binding domains, transmembrane segments, extracellular, intracellular loops, whatnot. So this just further suggests that we can try asking a lot of different questions of this data, but also that those questions should be very specific and that we should carefully consider specific phenotypes to navigate the complexity of the data. So the first question one might want to ask is, of course, about the disease liability of all the examined variants. Here on the right, we have just an illustration or reminder of what we are looking at. We have the reference trace from a wild-type Cftr protein, and we have the trace from Cftr with R117P variant in DMSO, not pretreated with any correctors. So the level past the four-scalene addition, as we discussed, is just a residual function of that mutant, which in this case appears to be about 10% of wild-type function, maybe just a bit more, at least in the particular experimental repeat we're looking at here. And here at the bottom, we show the residual functions of each of the studied variants. And we are going to see many plots of this type. So the location of each bar just shows the position of the corresponding variant on the Cftr protein sequence. And the height represents a particular metric, in this case, residual function of each variant as % wild type. You can see that we have a broad distribution, of course. Some variants have pretty high residual function. Maybe those are very poorly studied putative CF variants that were never confirmed and that in reality are not disease-causing. Maybe despite being located inside the exon, though their disease mechanism requires the native genomic context. Maybe they affect splicing, for instance. But then there are variants with some intermediate residual function that might be associated with milder form of disease. And then, of course, there are many variants with zero or near zero residual function. And these are the ones we want to zoom in onto. One has to start somewhere. So here we select only the variants with residual function less than 10% wild type. Those are located right here at the bottom of the graph below the blue dashed line, if you can see. And there are about 200 such variants in our data. And they're quite consistent between the independently performed studies 1 and 2, as this Venn diagram shows. What we can also see, however, even from here, is that the number of these molecular probes, well, it's large, and they are still distributed quite randomly and uniformly along the protein sequence if you look just at the guys below the blue dashed line. So let us try asking a more pointed question. Namely, let us restrict ourselves to those severe low residual function mutations that are likely to belong to the very specific classes of gating or conductance variants. We realize now that a variant might exhibit a combination of different mechanisms, but for the mutations that are squarely in one of those specific classes, by definition, the protein is produced, folded, trafficked to the membrane successfully. However, with the gating type mutation, the channel is just locked in the closed conformation. It can't open. And with the conductance type mutation, the channel might be able to open, but the ion flow through it is disrupted, for instance, by improper local fold or wrong polarity inside the channel, whatnot. So to favor such variants in our selection, in addition to residual function below 10% wild type, we require the protein to be produced, at least to some degree. So we use our Western blot data and require total protein and band C to B ratio to be at least above 30% or so of wild type. So it's not even a very stringent filter. But this requirement trims our selection very, very considerably, leaving us with 19 such residues selected repeatedly between study 1 and study 2. So we can immediately see that these variants are all internally positioned, in fact, and heavily clustered near the entrance to the channel or in the NBD domain. And also here we can see that they are all under the surface. By taking even closer look, we can see that those not this closer look, this one, we can see that those putatively gating or conductance severe disease variants are very heavily concentrated in a few regions which are known to be important for channel proper opening and conductance. So first, we have hotspots in ECL1, TM5, 6, and 8, and all 12 TMs are actually shown on this linear diagram as those additional yellowish bands. And one can see how, despite being very far from each other in linear sequence position, which probably doesn't matter much, they align against each other extremely tightly on the 3D structure. This upper structure diagram presents the view of the channel from the top, from the outside of the membrane and towards it. So this is the entrance to the channel right here, or one end of the channel at least. The red residues are those selected by our query. And you can see that ECL1 is right here, the usual suspect known to be very important, and TM8 aligns right against it. And a separate group of variants is found around G551 residue, of course, as well as around residues 1240 through 1250 approximately. And again, in the fold, those guys literally come together, they group together around the ATP binding site. Also, just as a sanity check, we definitely identify with our procedure, with our query, a number of residues that are known to cause specifically gating or conductance defects when they are mutated. Now, of course, we can ask ourselves, but what about those severe mutations that we did not select in the previous query? Well, we have a large number of them, and they seem to be located all over the protein. Admittedly, some of them are buried, some are on the surface, as shown here. We just draw here the surface of the protein, and those severe, less than 10% wild-type residual function variants highlighted with red. You see there's lots of them on the surface. It's still tempting to speculate what these residues exposed on the surface that they might impact co-translational folding, affect interactions with chaperones and whatnot, but we do not have any hard evidence for that, of course. And I can only add that it is also likely that we are simply dealing here with the fact that variants that are not specifically gating or conductance mutations are not in themselves in one or just two other specific classes of disease mechanisms. It's just everything else. There are potentially many different mechanisms responding hotspots, even if they exist, are just mixed together here, and also some mutations may exhibit multiple disease mechanisms at once. So basically back to the point that it is quite possible that more specific queries would help here too. And I will leave it at that for now. Instead, I'm going to switch to a different example. I'm going to look specifically at the response of different mutant Cftr proteins to the potentiator. And in order to quantify that response, we are going to do the following. As illustrated in these conductance traces, we are going to look at the increase of the conductance after addition of the Cftr on top of whatever residual function the variant might have had, or none for what it was. And I am going to represent this gain of conductance as the percentage of the total conductance, the sum of residual function and the gain. So basically, if there was no residual function whatsoever, then it will be 100% of the ratio. Everything is due to potentiator alone. This metric, of course, is only meaningful when at least some total response is present. So I'm going to select only the variants with total response, residual function plus Cfdr on top, exceeding 10% wild type. And since this is more of a structure-activity relation question before everything else, I'm going to include both untreated and corrector-treated samples. I'm literally interested here in all residues where Cfdr can readily potentiate the channel, regardless of how the channel protein got to the cell membrane in the first place. So the diagram on the right shows our chosen Cfdr potency metric on the y-axis for each variant with total response above 10%. And we can see from this plot already that while we have many residues that respond to Cfdr very readily, those higher green bars, they are not uniformly distributed and seem to start clustering in some specific locations. So let us perform a selection, and just strong responders, those that are above 50% for the ratio of Cfdr response to the total response. We get 59 residues that are repeatedly, consistently found in both studies, with each single study giving us about 70. And 19 of those overlap with the putatively gating variants that we selected and discussed earlier, which makes perfect sense. And we can also see that these strong responders are not all strongly clustered in one place anymore. But in addition to the usual suspects in the ECL1, for instance, they seem in general to be still located either inside of the channel or again in the NBD domains, not far from the ATP binding pockets. And one can see from the structure diagram that while we have many residues here on many different helices, they seem to be always located on the inward-looking turn. So again, it is feasible that they might be negatively affecting the ion flow, for instance, and keeping the channel open for a longer time might be really helpful. And that's why they respond so well to the potentiator. And to bring this to the end, I'm going to show one last story where we now look at the response of the specific Cftr inhibitor molecule, which, as you remember, we add towards the end of each conductance measurement run. So again, we define the response to inhibitor here as the magnitude of the signal drop after the addition, measured as the percentage of the total response before the addition. So for the full inhibition, which we do expect to occur, we expect the values close to 100%, some experimental noise and baseline drift notwithstanding, just as it is shown in the trace on the top. And then we noticed that a small number of variants showed no or almost no response to the inhibitor, as shown in this trace at the bottom. These observations are consistent across the sites, but they disappear in study 2, where we in fact use a combination of inhibitors, not just this inhibitor 172 molecule, but also another one in a cocktail. Again, in order to be able to determine the percentage of the total, we need the total signal to be present in the first place. So we choose all the variants untreated or pre-treated with corrector, where the total signal, residual function plus our Cftr on top exceeds 20%. And out of those, we select the variants where the signal drop due to the inhibitor is less than 50% of that total. There are only a few variants, 6 at only 5 different residues that are selected by this query. And on the protein sequence, they fall into both MSD domains. However, where do they get when the protein folds? On the 3D structure, these residuals perfectly align against each other, inward looking near the entrance of the channel. It is not as unfeasible that this could be the binding pocket for the inhibitor molecule. And as it binds to both MSDs that form the 2 opposite walls of the channel, it just renders the channel shut. It is also worth mentioning that at least one of these residuals was already shown previously to be required for the inhibitor to work. So with that, I would like now to conclude by reiterating that we have assessed the residual and treatment-induced function, as well as C and B band protein amounts in over 600 Cftr variants in 2 subsequent theratyping studies. And while this is neither the original nor the main purpose of our experiments, it just happened, and to the best of our knowledge, this makes it apparently the largest structure activity study conducted on Cftr. We just finished integrating the data across the sites. We are still identifying, removing the outliers, and we believe we just scratched the surface here, but our initial analysis appears to show that the data set can be useful for further querying structure-activity relationships and formulating hypotheses. And of course, what I'm presenting here comes from the joint work of a large and great team who all share in the outcome of the project, and in this project having happened in the first place. Thank you very much for your attention.
Welcome to the panel discussion of Symposium 18. We're delighted that you've joined us. You've listened to some fantastic talks. We're very excited about those talks, and I hope that you are also. So Dr. Hong from Emory and myself, David Shepherd from the University of Bristol, we're moderating the session. We've already got a few questions that have come in, and we would like to go round the panel with those questions, and please encourage you to sort of post further questions or to vote on other questions. So let's make the most of this 40-minute discussion. So Professor Chen, one question we already have is the pocket open and similar in size in the unbound open and closed structure? Or is there a conformational change / induced fit that needs to take place for corrector binding?
Yeah, it's a great question. We compared the structures carefully. We didn't see any conformational change in this pocket when you compare wild type Cftr in both NBD separated or NBD dimerized conformations. So it binds to a domain that's part of the rigid body movement during the conformational change. Drug binding did induce some local conformational change of 2 side chains, a lysine and arginine. So if that's the induced fit you're talking about.
Okay, so can I follow that up with the second question from the speaker, that Professor Platt? So what could be the function of the cavity in native Cftr? Can it bind lipids? Is it important for regulation? Is it interesting that evolution maintains such a destabilized pocket? So your opportunity to speculate.
Great question. I wondered about that myself. So first, does it bind to lipids? When we look at the structures of determining the absence of any of the correctors, if you go to very low signal level, which means you look at very noisy maps and see if anything could be there. At that level, you see noise everywhere, particularly in the transmembrane region. We do see some density that could correspond to lipids. So my guess is sometimes lipids do come in and help stabilize the protein, but the occupancy is very low compared to the drug. Why the Cftr maintains such a destabilizing feature? The same question can go to why the Cftr is so prone to mutations and cause disease. I wonder if at some point of doing evolution, there is some selective advantage to make this protein not very stable. There are different hypotheses, but I don't have a straight answer to that.
Thank you very much. Hanyi, can I now ask you a question from Professor John Hunt? Have you examined any delta RI constructs in similar experiments to assess whether Professor Govart's alternative folding of nucleotide binding domain 1 may be involved in Cfdr rundown?
Yeah, so great question. Thank you, Dr. Hunt. And yeah, I'm sure TC is watching, so hi. The answer is no, we haven't done experiments with delta RI. We do have those constructs. So great question. I'd be happy to experiment with those constructs. What we do know is that delta 5OA, which alters the structural integrity of MBD1, has severe rundown problems. So I would expect that anything that changes the structural integrity of MBD1 would have impact on rundown.
So, Hanyi, if I remember your manuscript correctly or your article correctly, you did also study the delta R construct.
Yes. So delta RI is the segmenting MBD1. We didn't do that, but we do have delta R construct, which is the complete deletion of the total R domain. This construct still run down. It's a bit worse than wild type, but not very much. So yeah, that's the question.
Okay. So can I then follow up with the question from Professor Plate then? So sorry, my tongue is twisted. Do rundown channels eventually recover in vivo? Is anything else missing for recovery, other protein interactions, or is the rundown state less critical in vivo given that ATP levels probably never drop below 10 micromole? That's a fascinating question, isn't it?
It is. So there are three questions. First one, do we see it recover in vivo? Well, I cannot answer to that because we did not do any in vivo experiments. In our hands is always patch clamp in vitro. We can follow that current for as long as 30 minutes. We do not see the current recover in terms of irreversible rundown. The second question is anything else missing for recovery? What we have is we add pKa back. So we try to control phosphorylation level. We show that it does not help recover the reversible or irreversible rundown. And also we just talk about delta R, which doesn't need pKa, is still rundowns. The third question, probably the most interesting one, does it have any relevance in physiological condition? First of all, you probably don't need to worry about wild type channels running down in physiological condition. But we do know that delta 5OA can run down in the presence of millimolar ATP. So you can do experiment, you add ATP, saturating concentration, you activate 5-OA, and you just sit and wait and see, and the current will just drop gradually. So with mutations that destabilize MBD dimerization or destabilize binding of ATP, you would expect to see rundown even in the presence of millimolar ATP.
Yeah. So, just to sort of follow that up, studying other Cftr variants, pathogenic variants, deactivation or rundown is a very frequent occurrence, and it makes studying variants very challenging.
Yeah.
I guess to sort of come back to the in vivo, it's not quite the sort of an answer to the question, but certainly the deactivation occurrence or the rundown of Cftr-mediated currents, it's not just a patch clamp phenomenon. When you do Ussing chamber recordings of Cftr-mediated currents, you would also see loss of delta F activity, particularly at physiological temperatures. So yes, it's a very challenging problem when we study Cftr function. Okay, so Professor Plate, let me try and scroll down to the question that came for you. Okay, so from Dr. Janet Allen, do you control for Cftr expression levels in your studies?
Yeah, that's of course a really important question, and I apologize if I didn't really specifically clarify how we sort of do the data processing. So of course we need to correct for different expression levels, especially in the absence of the corrector molecules. So you saw, I think in the Western blots clearly that the Cftr levels are quite different. So when we do the immunoprecipitations, we also recover different Cftr amounts. And that was apparent in the volcano plots a little bit as well. So then the first thing we do is, after we filter out confident interactors, we do a normalization to the bait Cfdr protein levels. And so then all of the interaction intensities are basically relative to a normalized Cfdr bait protein levels. And so that's important, of course, to quantify increased interaction changes with, for example, delta F and the destabilized variants to capture if the global protein levels are down.
Okay, Dr. Again, I've got my tongue twisted. It's easier. Pardon me, Andre, from Professor Plate then, have you considered machine learning approaches to predict impact of other substitutions? This should be a rich enough data set. But you're muted.
My apologies. Yes, that's a wonderful question. We didn't do anything to that effect so far, mainly because we are still finishing integration and cleaning up the data set. So the data you have seen are really, really recent. But we can try that. I think there are two ways to do it. One is to try to learn something about the sites, and as you probably imply with your question, predict function or effect of mutations on the other sites, which I think would be a little bit of an academic exercise, because in our list we already have pretty much every mutation known so far. But it's still certainly interesting. And at the same time, we could use types of models that allow for better interpretation of the effect of different variables, so that we could learn more about the actual drivers of the effects of those mutations. What is important? Is it a substitution? Is it change of the size of the side chain and whatnot, which might have more practical importance, in my opinion. But we certainly would like to try something like that.
Andre, can I follow that up with a further question? So this comes from Dr. Catherine Oliver. Can you describe other Cftr inhibitors that were used in your cocktail with Cfdr INH172, or my understanding is you only use Cfdr INH172?
In the first study, we used only 172, and that's where we have seen those differences. There were a few mutations that wouldn't respond. And in the second study, we used a cocktail of 172, and I'm afraid to be wrong. I believe it was GlyH101. I would need to double check.
Yeah.
But there were two, 172 plus something else. And in that case, all those mutations that didn't respond to inhibitor in the first study, they started responding. So it's kind of a control, if you wish.
Okay, so we've gone round once. So there were some further questions for Professor Chen, and so if I just try and come back up and find that. So this is from Professor Madden, Dean Madden. So this is both for Professor Chen and Professor He. So what are the prospects for visualizing the proposed ATP-1 bound closed structure? Could a combination of stabilizing, destabilizing interventions trap ATP-1 bound ATP-2? So let me say that again. Could a combination of stabilizing or destabilizing interventions trap the ATP-1 bound, so ATP bound at site 1, whereas ATP site 2 being empty structure? Is that sufficiently clear, or do you want me to try again?
I can try to answer. I definitely possible, and now I feel really motivated to try. So I can imagine we can make mutations, for example, to prevent ATP binding in site 2, or to strengthen ATP binding in site 1. The worry about introduce those perturbations is how to correlate the structure with the CO state. Probably, if I have to do it tomorrow, the first thing I would try is to determine the structure at low concentration, like say 10 micromolar ATP, like Han Yi presented. Yeah, it's definitely possible. I think the challenge is to correlate the structure with the functional state.
Absolutely. Han Yi,
Yeah.
did you want to add anything?
I would just add that I agree. It's hard to correlate. If you want to do mutations, you could probably try those that destabilize binding of ATP in site 2, maybe G551D, which would also be interesting for clinical relevance. And you use like high affinity ATP analog for site 1, so that site 1 can remain bound.
Yeah. Okay. Han Yi, Professor Hunt has a further question. So, what temperature do you use? And the comment is, even wild type MBD1 unfolds to a significant extent at 37 degrees, although very little below 30 degrees.
We use room temperature to do all the experiments.
Yeah, I thought that was the case, but it's good to sort of clarify that point. Okay. So please come in and ask further questions. Professor Plate, can I just, probably the answer to this question is no, but let's just try it. So you've looked at hyper-responsive variants, and you've looked at very unresponsive variants such as G85E. What about N1303K, which is very unresponsive, but is an absolutely fascinating variant for so many reasons?
Yeah, I mean, that one's, of course, a little bit more prevalent in patients, and it's also, I mean, extensively characterized what the quality control mechanisms were. And that's actually the DNA JB12, which I highlighted. I think Doug Sear's group just recently showed that's really critical to triage N1303K. So we haven't done experiments with it yet. So yeah, you're right about the no answer. But we've definitely considered adding that into our panel of variants to probe. And I think now that we can the TMT sets are now expanded too. So This was collected with an 11-plex data set where we can multiplex 11 conditions. Now it's up to 16 and 18 conditions as well. So the mass spec technology also is sort of increasing rapidly, and we're excited to kind of expand capabilities.
And can I sort of follow up then? So, you know, it was the absolutely fascinating data with both LumaCFDR and with Cfdr. Have you tried both together yet?
Yeah, so in the we've compared basically the Cfdr versus LumaCFDR or Cfdr response in this case actually, and then the combination. So my student Minsu Kim, who I've highlighted briefly sort of towards the end of the talk, he's giving another presentation this afternoon. So I really recommend you tune in for that if you like to see more.
We won't give away any of the punchline. We encourage everybody to wait until this afternoon or this evening in European time. Okay, so brilliant. Thanks so much. Andre, can I come back to you? And so you had a number of responders that got to 100%. So there were like 100% wild type response, and a couple of them were located in the R domain, and then with ivacaftor there were also sort of like 100% responders, and there was at least one in nucleotide binding domain 2. Are you able to identify those?
We certainly have the identities of each and every variant we studied. I think we should be very careful, especially with the variants that respond very strongly. All of them pretty much are extremely rare. There are no surprises, things that people know about, we know that they are disease causing. They're extremely rare, poorly characterized. And it is possible in each of those cases pretty much that despite the fact that it's located in the exon, so we thought we could assess it, it might be affecting splicing or whatnot. But we are definitely going to look at them closer one by one, but I don't have any specific comments at the time.
Great. Okay, so if we start going around again, so Professor Chen, so there's a question then. Would you expect any changes in the binding site for LumaCFDR when ivacaftor is added together, given the previous data about ivacaftor destabilizing the F508del when it was rescued with LumaCFDR?
Yeah, we analyzed the structure of the ivacaftor bound conformation, and we didn't see any conformational change in the binding pocket for LumaCFDR. So the effect observed functionally probably is not due to structure changes.
Right. Great. Thank you very much. So Hanyi, so from, and I apologize if I sort of make a murder the name. From Dr. Patel, so have you tried to see if any of the modulators help keep the channel open along with ATP? Have you tried to see if any modulators help keep the channel open along with ATP?
Yeah, great question. So we haven't done experiment specifically for modulators effect on rundown. But if we assume that having a dimerized MBDs or even just partially dimerized MBD will keep the channel happy, then you would expect that potentiators can help the channel stay healthy. However, I think it's from Dr. Shepard's own lab, rundown of 508del becomes even worse with ivacaftor. We don't know exactly why that happens. One of our hypotheses is that maybe when you have potentiators, you force the channel to gate more often. Every time it closes, you have this hydrolysis happening. And from our paper, we show that hydrolysis deficient mutant does not have irreversible rundown. So in other words, when you don't have hydrolysis, the channel somehow is resistant to rundown. This is purely speculation. We speculate that the the energy from hydrolysis sort of breaks the dimer apart. It hurts the dimer in some way. So there will be a delicate balance between the potentiators helping the channel stay open and the ATP hydrolysis that hurts the channel when it breaks the dimer apart.
Great, thank you very much. So for Lars and for Andre then, a further question from Dean Madden then. Would it be feasible or slash informative to apply multiplex mass spec approach to characterize a fair number of the miscellaneous, in inverted commas, surface mutants and see if patterns emerge in terms of their interactomes? So would it be feasible slash informative to apply the multiplex mass spec approach to characterize a fair number of the miscellaneous, in inverted commas, surface mutants and see if patterns emerge in terms of interactomes? I don't know what a fair number is here.
I can take part of that question. I am a computational person, so I would say it would be incredibly informative if feasible. So if we could do affinity tag mass spectrometry and just see what's pulled down, because those mutations on the surface, by all means, they could disrupt interactions with chaperones, and it would be nice to be able to see those. As for feasibility, I would leave it to experts.
Yeah, I mean, I have to just echo that. I mean, I'm really excited to see these data sets, and we've definitely, like I said, we already are planning for expanding this to other variants. So I think we should definitely touch base, and I would love to kind of like prioritize at least. I mean, definitely not all 600 is really feasible, but even if it's just starting with a dozen or expanding from there, that would be informative. And the one thing that I will add to this too, even it's not just surface residues, of course, that will actually influence the proteostasis interactions. Like even buried residues that are sort of influencing the folding trajectories are, of course, really critical, right? So those may be quite informative as well to kind of correlate both correctability to structural stability of the protein to then the proteostasis interactions that really guide the quality control for Cfdr.
Great. Hanyi, you're very popular. So there's a couple of questions. So from Janet Allen is asking, does the R domain play any role in stability of Cfdr and the sorry, I've just had my chat move. And the dynamics of the channel. So does the R domain play any role in the stability of Cfdr and the dynamics of the channel?
Okay, great question. And I would also like to hear from Dr. Chan's opinion on this question. We do speculate in our paper that the R domain, because it connects MBD1 to TMD2, it was sort of helps the channel to stay healthy in some way, to prevent this further separation of the two halves of the channels. So yeah, I would say it definitely worth experiments to try different partial delta R and see whether they round down more easily or they are tough. Yeah, that would be my answer.
Okay, and then there's a further, I think this is a further question directed at, oh, sorry. So Professor Chen, did you want to sort of come back or?
Yeah, I agree. I think R domain definitely plays a role in stability, probably as well as dynamics of the channel. From TC's own data, I think he showed partially phosphorylate. It's whether depends on how much you phosphorylate R domain, the channel gates quite differently. Also, there are early data showing people use constructs deleting with the R domain delete, but when you trans complement R domain, the channel is more stable.
Yeah. Okay, so Hanyi, then this further question, anonymous. So G551D behaves like a rundown channel in our Ussing chamber experiments. Could Cfdr recover irreversible rundown in negating mutation?
So I'm curious about what the behaves like a rundown channel mean. In our hands, every time we do G551D, because it's excites patch, the current is very small, so it's very hard to quantify the reversible, irreversible rundown thing. And the second question, could Cfdr recover the irreversible rundown in a gating mutation? So that's something we can test. I don't know the answer yet. But sorry.
No, I was agreeing with you. Sorry.
Yeah, we can make the channel rundown and just add Cfdr alone and see what happens afterwards.
Yeah, so I could add that our experience of studying G551D, so we would typically work at 37 degrees C, and there really is a striking difference compared to studying F508del at 37 degrees C. So the G551D channel, yes, its activity is very low compared to other variants and particularly wild type, but it does remain active over considerable periods of time, providing your recordings last those time periods. Whereas F508del, you have a very short window of opportunity to study the channel at 37 degrees C. And then there was some very interesting data from Deborah Colon and her colleagues in their Cfdr paper, suggesting that the treatment of G551D with Cfdr sort of made the structure more flexible, whereas for F508del you were destabilizing an already unstable structure. Lars, a further question from Catherine Oliver. It's interesting that certain cellular factors are enriched for interaction with F508del and P67L, but then those interactions are decreased in response to corrector treatment. Have you tried inhibiting any of these factors to assess whether biogenesis of mutant Cfdr is improved? That was a mouthful. Do you want me to try that again?
No, it's fine. I caught the question if the audience did as well. Yeah, so the question is, of course, like now address, or you leveraging this data set to actually propose new correction strategies targeting these proteostasis factors. And yeah, I mean, we're certainly not the first ones to go after this, and I think that's been shown with quite a few sort of central chaperones, co-chaperones, and other factors that knock down siRNA-mediated knockdown, for example, can rescue delta F, 508 trafficking, and also other mutations. So we do identify some of these, and we've kind of cross-checked our data set with those discoveries. So, for example, I think BAC2, we highlighted DNA JB12. I already talked about Durlin1, Park7 were some that I remember now, that where the knockdowns are well validated to increase trafficking. We haven't yet gone sort of beyond that and tried the interesting new ones that we've identified, and specifically also ones that are now different between the hyper-responsive and the non or poor responsive variants, to kind of ask the question, like, how are these actually involved in dictating the difference in the response. But one complication always is that these are highly coordinated, right? I like to highlight that these pathways aren't, the protein gets handed off from one chaperone to another proteostasis factors, and there's coordination that's required, and they're highly redundant. So when you knock down one, then another pathway might take over.
Okay. Andre, can I come back to you? So the Cfdr INH172 data are fascinating to see, and it's great to see the sort of the agreement with the data from Luis Gaglietti's group. So Paola Vergani was in her recent review with Hugo de Jong was she was proposing, so that review was proposing that Cfdr INH172 is sort of plugging the intracellular portal. Is that correct, or is that location too low compared to where your variants are located?
I believe our variants are located a little bit higher. They are really near the mouth of the port. Yes.
Yeah. So please correct me then. So do you think then that Cfdr INH172 goes through the portal and then is plugging the intracellular vestibule, or is it, because it's an allosteric mechanism, it's not an open channel blocker. So is it sort of, it's not in the permeation pathway to target that site?
If I understand your question correctly, I think that it's only one possibility, not necessarily the top one, that is the binding site. Maybe not. And at the same time, the actual binding site, we are limited to the mutations that are in our data set. It might be that we just don't happen to have the probes right where it actually binds, while the site that we detect, well, it affects the inhibition, but it's not the binding site.
Yeah, exactly. Yeah. No, but it's fascinating to see the data. Yeah. So thank you. So Professor Chen, can I come? So we haven't got any further questions coming up yet in the chat, or in the Slido. But can I ask about Cftr? And this may be something that's sort of more a speculative question rather than something based on data. So there are some very interesting data now with Cftr saying that it has both corrector activity and potentiator activity. So I remember from Professor Werkman's early work on these dual acting molecules that one idea might be that you've got one binding site for correction, a second binding site for potentiation, or the alternative idea that you've got a single binding site where both activities take place from. Do you have any thoughts, or is it still too early to comment on this?
Yeah, no, we did also see potentiation in our own hands. We're really interested to understand that. I think both possibilities are possible, but if I had to guess, I would think if there is a binding site that can stabilize the protein in the active conformation, or elongates the lifetime of the protein during folding, it could potentially have both effects just through one binding site. Yeah, of course we have to see. Yeah, it's a little early to answer
but the data are absolutely fascinating. That you've got Cftr acting as a dual acting molecule. Yeah, really fascinating. Yeah.
Yes. Yeah.
So much more to be done with with Cfdr modulators. Yes. Yeah. We've still got time. So so you sort of touched upon the idea in your sort of previous the previous question. So Hanyi, do you think that small molecules that target site 1 or somewhere in the region of site 1 could have sort of therapeutic potential, or is that site going to be, because it's going to have overlap with other ATP binding proteins, it wouldn't be a suitable site for trying to find modulators?
Well, okay, purely speculation. I certainly hope that there will be modulators that stabilize MBDs. Now we have Cftr, the Galapagos compound. They all work in the transmembrane domains. Of course, we don't know where Cftr works, maybe in the MBDs, but I would expect that when you have some drugs that stabilize MBDs, particularly Cy1, binding of ATP in Cy1, you can have sort of synergistic effects on top of either Cftr, because it will both be a potentiator and like a stabilizer. It stabilizes the channel in memory.
Yeah. So I think that having those types of molecules available that stabilize the protein, I think those are going to be very valuable resources. So yeah. It's just how to, what sort of assays do we need in order to effectively discover those types of small molecules? Yeah, I think that's going to be the challenge. So we've got a couple of minutes left. Does any of the panel want to ask any of the other members of the panel something quick?
I have a quick question for Henry. Yeah, a great paper and great talk. The question I have is the slow component tau2. Why would that number change? So tau2 increase as you increase the washout time. Do you have explanation with why it changes?
Yeah, so we have this proposed mechanism that you go from a closed state with ATP inside one to the closed state, the reversible closed state, where the two MBDs are already separated. And if that counts as one step, then potentially you can have multiple steps that you go further into those round down states where the MBDs are separated with different distance. So the longer you wash out, the more channels go further into those deep round down states. And for those channels, you're going to have to wait longer for them to come back to the open state. Like you go from step 1 to step 2 with 5 seconds washout of ATP. If you wash out for 50 seconds, those channels might go further down to other round down states. And those could potentially be, all of them could be reversible. It's just that the time for them to go back to the open state is much longer. So the tau2, the recovery from reversible will be prolonged with longer washout. I hope that is clear.
Just a follow-up. So the time scale for protein conformation or change usually is microseconds or maybe sub-seconds. So the tau2 is minutes. Mm-hmm. That just makes me a little surprised.
Yeah, we will. That's just our observations, and we are not sure if that scheme, that mechanism we propose is correct. We can't say it's correct, and we certainly hope to see different structures, like Gaussian structures, that can support these data.
Okay, so I think we're, oh, no, sorry. So Janet Allen has come in with another question, so please, quick. The ATP binding pocket of kinases have generated thousands of ATP analogs with high affinity binding as well as block kinases, pretty well characterized in terms of binding to the pocket. Could any of these be used to answer the question? The ATP binding pocket of kinases have generated thousands of ATP analogs with high affinity binding as to block kinases, pretty well characterized in terms of binding to the pocket. Could any of these be used to answer the question? I think that's great food for thought. So we need to find some collaborations with industry then. I think that's the answer there. Okay, I'm conscious of the time. I just want to say, we've had such a fantastic Symposium 18, really wonderful presentations about structure, function, and interacting networks, and lots and lots of variants and their impact on Cfdr structure. So to our speakers, thank you so very much indeed. To the audience, although it's all virtual, thank you so much for your interest in this symposium. We greatly appreciate that. And I'd like to just finally thank my co-chair for keeping me going, providing me with questions. And so John, thank you so much. And and the support that we've had from Zachary and Kirsten. So thank you so much, everybody. It's been a wonderful session, and I hope you enjoy the rest of the meeting. Thank you very much indeed.
Thank you.